Hi, I'm Mike Larson. I'm going to be talking about developing an H&E alternative using approved drugs as off-label fluorescent contrast agents for intravital microscopy or optical biopsies. We perform many image-guided biopsies in the medical imaging department, and they are prone to problems, including risks of complications, lack of feedback, and there's been an explosion in targeted therapies and their prerequisite companion diagnostics. Unfortunately, the small volume from needle biopsies is insufficient for companion diagnostics about a fifth of the time, and therefore patients don't get their targeted therapy. Additionally, needle biopsies are subject to sampling errors due to Darwinian-like evolution with different genetic mutations within a single tumor, so that a single sample doesn't fully represent the whole. Thus, needle biopsies have been called the limiting factor in precision cancer care. Intravital microscopy or optical biopsies is a solution. It actually puts the microscope into the patient so that we can get subcellular resolution, but without the penetration limitation. And unlike typical needle biopsies on the left, which disrupt tissue and lack a cellular level real-time feedback, optical biopsies on the right could provide or minimize the tissue disruption and provide that real-time cellular level feedback. But to improve upon the optical technology, like with physical biopsies, we need to add stains or dyes to improve tissue contrast, like with white bright light or bright field microscopy or fluorescence microscopy. So the objective of my work was to develop a hematoxylin and eosin or H&E alternative using approved clinical drugs as off-label fluorescent contrast agents to better enable this intravital microscopy or optical biopsy technology. The first portion of my work was to develop a list of DNA binding or hematoxylin alternative and cytoplasmic or nonspecific protein binding eosin alternative ex vivo or research molecules and test them for structural similarity against clinical or in vivo used drugs. I used a fairly comprehensive drug database that allowed a structural similarity search. So this is a research dye that stains DNA, and I put that in, and output was structurally similar approved clinical drugs. In doing that, I was able to come up with a list of over 200 structurally similar drugs to any of the 50 research dyes I tested. The second portion of my work was to test some of these DNA binding or protein binding drug combinations in vitro for fluorescence compatibility. This is an example spectra, fluorescence emission spectra of some of the drug dye combinations where I have in red the DNA binding drug, the blue is the nonspecific protein binding drug, and the combination I can still see two distinct colors even with a single laser excitation. This is a table summarizing some of my drug dye combinations and the expected colors. The third portion of my work was to test these combinations on unprocessed ex vivo lung samples using a single laser excitation confocal microscopy. On the left is one of the drug dye combinations, with the blue being the nucleus and the green and kind of red being protein. And on the right is a H&E for a qualitative comparison. And to make that a more intuitive comparison, I inverted the hematoxylin eosin on the right, and while I'm not a pathologist, I do think I'm getting close. And here's another example of a different drug dye combination. These are a list of the top drug dyes and proposed intralesional dose for optical biopsies and how it would be only a fraction of the typical clinical dose. In conclusion, I've demonstrated some qualitatively off-label drug dyes as H&E alternatives for optical biopsies or intravital microscopy, and have lots of ideas planned for the future. With that, I'd like to thank you for your time and thank my mentors for their help.